A very good morning. So, today we are going to study an important topic of chromosome, that is structure of chromosomes. So, this gross structure of metaphase chromosome may be studied with reference to the following parts. So, first is a chromatids. It is a part in the chromosome which is having a two cylindrical rod shaped compartments of a metaphase chromosome. So each is called a chromatid or a sister chromatid. The two are produced as a result of replication of a single chromosomes. Hence they have a identical structure. They are held together at a point called centromere. So here chromatid nothing but it is a two cylindrical rod shaped compartments, right, which is seen in the metaphase of chromosome, and it is called as a sister chromatid or a chromatid, right, and the two are produced as a result of replication of single chromosomes. Right, next part is a primary construction. It is the region where the chromatid is comparatively narrow than in the remaining part. In this stains, in this region, in the primary construction region, which is narrow, right, and it is lightly stained, right, and it is called the primary construction, or it is also called as a centromere. The centromere is not located in the same place in all chromosome. So in the last session we discussed about the place of the centromere depends upon the shape of the chromosome, right? So it may be in the middle, or it may be one side of the midpoint, or it may even be at the end of the chromosome. The difference in position is the basis for classification of chromosomes. So here next kinetochore, another third important part. So the kinetochore is the complex and a specialized region associated with the centromere. It is made up of protein complexes. In mammalian chromosomes, it is lamellated and composed of three plate-like structures. The kinetochores offer two chromatids face opposite poles. They are sites where spindle fibers or a kinetochore microtubules, that is nothing but a important part here, become attached and pull the sister chromatids to opposite pole during anaphase here, right. The number of kinetochore microtubules that attach to each kinetochore differ in different species. It's not same in all species, right. In the yeast only one attached, right. In the plant it may be 70 to 150 kinetochores are attached here. And next is a secondary construction. So the secondary construction is another narrow region like a primary construction on a chromosome. It is right, frequently lightly staining. It is visible under a light microscope. It also occupies a definite position. Right, all chromosomes of a nucleus do not possess secondary construction. Only in a few, like in human chromosome number 13, 14, 15, 21 and 22 possess secondary construction. When such a construction is present, it is usually associated with a nucleolus. In such chromosomes, the secondary constructions are called nucleolar organizing region, that is NOR. Right? And there are two NORs per nucleus, one each on the two members are a homologous pair of chromosomes. So next, satellite body, which is present next to the secondary construction, right. It is the bulb like terminal part of the chromosome. It extends beyond the secondary construction. It is also called as a turbinate here. And next part is telomere. The telomere represents the tip of a linear chromosome, one at each end. It has a special repeat DNA sequences. It prevents the adhesion of a one chromosome to another at the ends. So, a telomere is necessary for the proper replication of a chromosome. It helps in the attachment of a chromosomes to the nuclear lamina. Right. Next, chromonema. It is the thread-like part composing the chromosome. It is also called chromatin fiber, and it is in an extended or a diffuse state within the nucleus when the cell is not dividing, right? But if the cell is dividing, the chromonema undergoes coiling to produce a visible chromosome, that is chromatid. So each chromatid has a single chromonema since it is composed of a nucleic acid, that is DNA, and combined with the proteins. So it is a nucleoprotein fiber and it is about 13 nanometer in diameter, that is chromonema. So here you can see the normal structure of a chromosome, which is having a two rod shaped chromatids, right, which is having a center or any other region primary construction, and at a one point it is having secondary construction. Above the secondary construction are a bulb like structure called as a satellite. On the top of the satellite you are having telomere. So next, chromomeres, another part in the chromosome. They are regions of the chromonema which appear like a beads under the light microscope. Each is a tightly coiled region of a chromonema. Chromatin, and another important part here. The word chromatin refers to the stainable material composing the fibrous unit of a chromosome or the chromatin fiber. If the chromatin fiber densely coiled and folded or condensed, it takes more stain and appears darker. We call that as a it is heterochromatin, right? If it is a condensed, densely coiled, and takes more stain and appear darker. If it is diffuse or a long, thin or extended, it takes less stain and appears lighter. Right. So here the condensed state, that is darkly stained, we call heterochromatin, and the diffuse state, that is lightly stained, is a euchromatin. The euchromatin can condense to become heterochromatin and vice versa here. So this is about the structure of chromosome. Now we discuss about SAT chromosome, another important chromosome. It is a type of chromosome here. SAT chromosome nothing but stands for cyanoacidothionucleone. The term applies to all satellite chromosomes and to those secondarily constructed chromosomes which are considered in the origin of a nucleolus. While studying structure of chromosome, I told you secondary construction, satellite body, nothing but a SAT chromosome. It is not present in all chromosomes, right? So here in general, it applies the chromosomes associated with the formation of a nucleolus. So the satellite chromosome or a SAT chromosome has a chromosome segment that is separated from the main body of the chromosome by such a secondary construction. The term is due to Sergei Navshin in 1912. The satellite at metaphase appears to be attached to the rest of the body of chromosomes by a thread of chromatin. Point 4. Sorry, 4. So there are four main types of chromosomes here. metacentric, submetacentric, acrocentric and telocentric. So you know, right? So in human, each cell normally contain 23 pairs of chromosomes for a total of 46. 22 of these pairs called autosomes look the same in both males and females. So here you can see this structure of SAT chromosome, that is satellite chromosome, where you find a secondary construction. Above that you are in satellite structure, bulb like. So satellite chromosomes are a chromosomes that contain secondary construction or that serve as a identifying marker. Besides the centromere, one or more secondary constructions can also be observed in some chromosomes. At metaphase, this is the case with the acrocentric chromosomes, that is 13, 14, 15, 20 and 22 chromosome, which contain a segment that is separated from the main body of the chromosome by a such secondary construction. The secondary constructions are always constant in their positions and hence can be used as a markers to identify these particular chromosomes. The Y chromosome can also contain satellites, although these are thought to be translocations from autosomes. And the chromosomes containing satellites are called a SAT chromosome. So here we discussed both important topic, that is a normal structure of a chromosome and a type of chromosome, that is a SAT chromosome, which we also call as a satellite chromosome here. Thank you.